Today we're going to talk about the exciting field of stem cell biology research. It's a field that has completely exploded in the 21st century. It's given rise to fields of study that didn't even exist a few years ago. Fields of study such as regenerative medicine, cellular-based therapies, and these are incredibly important fields of study for people with degenerative and genetic diseases. The outcomes from these studies are going to have a huge impact on people who are affected by these types of health conditions. And in fact, Canada has played an enormous role in the study of stem cell biologies. Right here in Toronto is where stem cells were discovered. They were discovered in 1960 by Drs. Till and McCulloch, and the discoveries and research going on in Canada has continued to grow. In 1992, adult neural stem cells were discovered at the University of Calgary by Dr. Weiss. Up to this point, it wasn't even known that adult neural stem cells existed, and this has huge potential for people with brain and spinal cord injuries. In 2011, just to name one other example, there are many examples. In 2011, Dr. van der Kooi, again here in Toronto, discovered pancreatic stem cells, and these pancreatic stem cells are actually capable of producing insulin, and this, as you can imagine, is of huge importance to people with diabetes. So not only in Canada have there been enormous discoveries in stem cell biology and potential stem cell therapies, there have been many media reports that you may have seen over the past few years and months about discoveries and apparent breakthroughs in other places around the world. Is this hype? Is this hope? Because some of these headlines kind of seem a little bit too good to be true. Well, the purpose of this module will be to help you through your basic conceptual understanding of cell and molecular biology, together with what we're going to talk about today, begin to make sense of these media reports, not just the ones coming now, but the ones that you're going to see beyond now when the course is over. And like the cancer module, you're going to need to apply what we learned to what we're going to talk about in this module. And so how we're going to do that is we're going to talk about what stem cells are, so the basic biology, some definitions. We're going to talk about the different types of stem cells, which is actually more exciting than it sounds, because the different types of stem cells are fairly recent discoveries, and I think you're going to find that quite interesting. And then we're going to conclude with an overview of current and future stem cell therapies. The potential of stem cells, I can't really overestimate it. It's actually absolutely breathtaking in terms of the quality of life and the treatment for people with many types of diseases. And well beyond this course, you're going to be feeling the impact of stem cell biology. Not only will you have a say in the type of research and therapies that will be offered, but it is undoubtedly going to impact either you yourself during your lifetime or that of the people that you are close to. So let's get started. So what exactly are stem cells? Well, before we get to that, let's start off with a definition. Differentiation occurs when a cell or a group of cells becomes more specialized in structure or function. And this is an important concept. Here we have a cell, it's called a precursor cell, and through the process of differentiation, so this is one type of cell, a precursor cell, it can give rise to very different types of cell types. It could be fat cells, it could be bone cells, could be white blood cells. In many cases, it can essentially be any of the cell types that are needed in the body. And the process through which that happens is known as differentiation. And these dotted arrows indicate that there are many cell divisions and steps that take place. Now, stem cells are particularly interesting because they give rise to all the different cell types that are needed to form our tissues and organs. So for example, this stem cell has gone through a cell division, producing one stem cell plus a precursor cell, and this precursor cell, through the process of differentiation, is able to give rise to all these different cell types. Now, these are just three examples. Essentially, all cell types can arise from stem cells. And stem cells can divide to form two new stem cells, which is one thing that can happen. In this example, you can see a stem cell dividing into another stem cell plus a precursor cell, or a stem cell can divide into two of these precursor cells. So cast your mind back to mitosis way back at the beginning of the course, and this will look a little bit familiar to you. Precursor cells can change or differentiate again, just to review, into a new type of cell. Now, what's interesting, and you may not have realized, is that in the body, once differentiation has occurred, so once a cell has terminally differentiated into a fat cell, it cannot usually in the body change back into a precursor cell, okay? So this is a one-way, in the body at least, is a one-way direction. So a fat cell does not go back to being a precursor cell, and a precursor cell in the body does not go back to being a stem cell. So that's quite important. Now we're ready to talk about what stem cells actually are. There are three key properties, a couple of which I've already alluded to earlier, but let's go through them one by one. The first one is that they are unspecialized. So here we have a stem cell. It's considered to be unspecialized, and what that means is it does not have a tissue-specific function. For example, this stem cell could not carry oxygen like a red blood cell could. It can't function within your muscle tissue to move your leg, for example. So it's an example of an unspecialized cell, and it doesn't have tissue-specific functions. It has other functions, as we have seen and will continue to see, but it's not a specialized cell. Okay, it's unspecialized. It's capable of dividing and renewing itself for long periods of time, at least for the life of the organism. So here we have an example of a stem cell going through mitosis, giving rise to two daughter cells, one of which is the precursor cell that we talked about in the last slide, but the other daughter cell is a stem cell, and it stays a stem cell. Now this is an example of self-renewal. So we're not losing this stem cell when it divides and gives rise to the precursor cell, which will eventually give rise to all these various other kinds of cells. You still have a stem cell. You haven't gone down in number because of this self-renewal process, where during the mitotic cell division, one of the daughter cells stays a stem cell. So that's a process of self-renewal, and that's really important because we need to have some stem cells throughout the life of the organism. And then the third characteristic, which we have talked about, but I'll just review it with you again here, is that stem cells can give rise to specialized cell types, and these enable the body to grow and develop, as in an embryo becoming a fetus, becoming a human baby, and also in adults and children as well. Stem cells give rise to specialized cell types that allow repair and renewal of worn out tissues or damaged tissues. So just to review, here we have a stem cell. It can undergo a cell division, and we will retain an unspecialized cell, which is this daughter stem cell, but it can also give rise to the precursor cell, which through the process of differentiation, and remember I said these dotted lines indicate that there are many, many steps in cell divisions and changes in the cells that have to take place before this precursor cell could give rise to fat cells or bone cells or white blood cells, because these are terminally differentiated cells. And just to review, these terminally differentiated cells, which means this is their end specialization. They will not become anything except a fat cell or a bone cell or a particular type of white blood cell. That's what terminal differentiation means. These terminally differentiated cells cannot usually go backwards and change back into stem cells in the body. So now let's take a look at a really well-known example of differentiation in stem cells. This is in your bone marrow. This is what's happening in your bone marrow. So there are bone marrow stem cells, and after cell division, it gives rise to two types of what you might think of as precursor stem cells, okay? And With differentiation, for example, the lymphoid stem cells here, there could be many, many cell divisions and steps here. It would be better if in this figure they had shown it as a dotted line. Eventually, lymphoid stem cells and their precursors will give rise to different types of lymphocytes. Okay, so many cell divisions and steps happening here. And the same thing here. Here we have the stem cells giving rise to two different types of precursor stem cells, the lymphoid and the myeloid. And the myeloid, through much differentiation and many cell divisions, as I said, gives rise to all these other different types of blood cells. Now, just to give you an example, so after a lot of cell division and differentiation, imagine we get to the point where we are going to be making red blood cells, which are erythrocytes. Just cast your mind back to mitosis and remember that you really can get a lot of cells from cell division. So imagine, so this is your precursor for your erythrocytes. It divides, you get a couple more cells, these divide, you get more, and it keeps on going. And you can imagine this going on hundreds and hundreds, thousands of times within your body all the time. So the impression I'm trying to give here is we are only showing in this figure, you know, just one example of, and I don't want it to give the impression that it's just a couple of cell divisions. It's many, many cell divisions, so that you have lots and lots of red blood cells, lots and lots of lymphocytes, all coming from a single stem cell. Okay, so a single stem cell gives rise to many progenitors or precursor cells over the lifetime of the organism, and these in turn give rise to many, many, many lymphocytes, many, many of these myeloid type blood cells as well. Let's take another look at a common behavior of stem cells. Remember, we said that stem cells are self-renewing, so here we have an example of a stem cell. It's dividing. One of the daughter cells is remaining a stem cell and going through that self-renewal process that we talked about before, and the other cell is terminally differentiating into another type of body cell, you know, a bone cell or a blood cell. So this is one example. In the case where a stem cell divides and one of the daughter cells differentiates, what factors cause this? Well, it turns out it's very closely related to gene regulation, which we talked about in an early module. And the gene regulation is affected by cues from the environment or asymmetrical distribution of proteins or factors. So if we're talking about the cues from the environment, you can imagine a situation where we have a stem cell. It divides, the same DNA on each side of this dividing stem cell and the same DNA in each of these daughter cells. But one of them becomes a replacement for the stem cell and the other one differentiates. What happens is that this cell has encountered an environment that has cues that affect the gene regulation within the cell. So the DNA is the same, but the way the DNA is being regulated is different between these cells. And it's this effect on gene regulation that causes this cell to go through multiple changes and cell divisions that eventually results in a terminally differentiated cell. The other factor that can also be involved is the concept of divisional asymmetry, and essentially what this is an asymmetrical distribution of proteins or factors. So let's take a closer look. Here we have a stem cell which will ultimately divide into two daughter cells, again the same DNA, and these two daughter cells have the same DNA. But the difference is in this original stem cell, some special proteins went to one side of the stem cell, and they signal here that this new daughter cell should stay a stem cell. This cell over here, this other daughter cell, has the same DNA again as daughter cell number one. However, it doesn't have the proteins, it has other proteins, but it doesn't have these particular proteins that they're showing with this red line there. It doesn't have those particular proteins expressed that will tell it to stay a stem cell. And that affects the regulation of the genes in this cell. And this is what ultimately, after much gene regulation, many cell divisions, will result in a terminally differentiated cell. So remember, when the cell divides, the daughter cells have essentially the same DNA. It's how the genes in that DNA are regulated, and that is a response from environmental cues, or could be a result of the asymmetrical distribution of special proteins. And it is that gene regulation that will determine what the stem cell does. Okay, so I really want to emphasize this really shows the importance of gene regulation, and this is important from both cues from the internal environment, as you get the internal environment of the cell, as you get with divisional asymmetry, because you can see the environment within the cell is different on both sides of the cells, or cues from the external environment, and these affect gene regulation, and they therefore affect the organism right from the start. Now let's take a look at examples of types of stem cells. Here we have a human egg and a human sperm cell. When the egg is fertilized, you get a zygote, which is another name for the fertilized egg. And this is the ultimately powerful stem cell. This single cell gives rise to all the cells and all the tissues and organs that will ever be needed in the human body. And you can see as we go along with this arrow here, you can see here we have the developing embryo. There are stem cells right in the middle here. This is called the internal cell mass. And each one of these cells is capable of generating all the embryonic cell types in the body. So each one of these cells is capable of giving rise to all the tissues that the baby will ever need. And in this example here, they're showing you how it can take place in the laboratory. Is if a single cell is removed from this embryo. Now this can happen without destroying the embryo, and happens with embryos that are part of in vitro fertilization programs, and it's done with the permission of the donors. And so one of these cells can be removed. They can be cultured in the laboratory and under different cultural conditions, which, remember, that is equivalent to the environmental cues that we were talking about on the previous slide. One of these cells can generate more cells. You differ the culture conditions, which is essentially the environmental conditions, and from this single cell, you can generate many differentiated types of cells, liver cells, nerve cells, and so on. Also, we have a situation where this embryo develops into an adult. Well, the adult actually will have some stem cells, and so here's an example that we've talked about before, and this is the bone marrow, so there are stem cells in the bone marrow. Well, in the laboratory, you can remove some of these stem cells from the bone marrow, culture them, so they will multiply, you'll get more of them. Under different cultural conditions, you can get the stem cells from the bone marrow to differentiate into various blood cell types. But notice, the cells that came from this bone marrow were stem cells for making blood cells, also called hematopoietic cells. We'll talk about that in a further example, so you can see how it's spelled. But notice that the stem cells from here only give rise to blood cells. They do not give rise to nerve cells or liver cells. Okay, so just to review, there are two categories of naturally occurring stem cells in the body. The first is adult stem cells, and these are found in adult tissues. These are responsible for repairing and renewing adult tissues. So if there's some disease or some damage or just normal wear and tear, these stem cells are responsible for giving rise to the differentiated cells that are needed to keep an adult body functioning in a healthy manner. The second category of naturally occurring stem cells are the embryonic stem cells. Remember I said these are inside the embryo here. They're in the inner cell mass. This whole structure is called a blastocyst, and that's the structure that's formed as the embryo is developed. These stem cells are very powerful and can develop into all the types of cells that are needed in a baby's body. The zygote, the fertilized egg, just going back a further step, this is the most powerful stem cell because it can differentiate into everything, both the things needed by the developing baby plus some of the extra embryonic materials that are needed, and we'll talk about that in a little bit more detail in a later slide. So right now, let's just take a look at an example of an adult stem cell that you're quite familiar with. We've seen this slide before. Essentially, these are the stem cells that are found in the bone marrow that give rise to the blood cells. Okay, and so there are definitely stem cells in your bone marrow, and these are the hematopoietic stem cells I was talking about. Basically, hematopoietic refers to the fact that these stem cells give rise to the various types of cells that are needed in your blood. And these stem cells exist in adult bone marrow throughout the life of the person, because you are always going to be needing lymphocytic type blood cells as well as the myeloid type of blood cells throughout your life. And in fact, and many of you will have already heard of this, bone marrow transplantation is a very well established stem cell therapy. It's been used for more than 40 years. We're going to talk about that in a little bit more detail later as well. So adult stem cells have been found in the bone marrow, and we've known about them for a really long time, but they've also been found in other tissues in the adult body. This is just a partial list. And while they've been discovered in many adult tissues, the thing that you need to understand is that the frequency of these stem cells within those adult tissues is fairly low, certainly compared to in the embryo. So these adult stem cells they're not all that easy to find. There aren't that many of them, but they certainly do exist. And for example, you can find stem cells, heart stem cells, that give rise to the types of specialized cells that are needed for good function of the heart. You can find liver stem cells, and those liver stem cells give rise to cells that are associated with the liver. The thing to notice about adult stem cells is that they tend to give rise to a limited set of types of cells, usually those associated with the tissue or organ from which they came from. Okay, so a little bit different from embryonic stem cells that can give rise to all the tissues and organs that are needed. Liver stem cells tend to give rise to the types of cells needed in the liver. Another important thing about adult stem cells is the fact that they can remain quiescent, which means non-dividing. You can think of that as meaning like quiet for very long periods of time until they're activated by a normal need for more cells, as I said before, to maintain tissues that have worn out or as a result of disease or tissue injury. And this is really important that they have this ability, is because you need these stem cells to be functional throughout the lifetime of the organism. And as it turns out, this quality of adult stem cells makes them quite important for research and future stem cell therapies as well. Now let's take a look at the most powerful of the stem cells, and these are the ones that arise from the embryo. So here we have a cartoon figure. We have an egg or oocyte and a sperm, and when that egg is fertilized, we have what is called a zygote, and this fertilized egg will undergo a few cell divisions, but notice it's still a single cell. There are some divisions, but it hasn't divided into multiple cells yet. And at this point, this fertilized egg, it's called a morula. You don't need to remember that. The important thing is you remember that it is totipotent, okay? Totipotent. And what that means is this is the all-powerful stem cell, what we have right here, this fertilized egg. It's totipotent because it can give rise to everything that an embryo will need to develop into a human fetus. And so it can give rise to the stem cells and the tissues that eventually will make the baby, but also those embryonic materials that I was talking about in the last slide. The totipotent state here can give rise to the extra embryonic tissues that are needed, such as the placenta. So remember, the placenta is where the baby gets nourishment from the mother. It's not actually part of the baby's organs, and it is through the fact that this morula is totipotent can give rise to tissues such as the placenta, which are extra embryonic, as well as the tissues and organs of the developing baby. So here we have this totipotent state. After some actual cell divisions, we get a whole bunch of cells. It's these outer cells that will go on and give rise to the placenta. But this inner cell mass that we talked about in the previous slide, it's the cells in the inner cell mass that will go on to give rise to all the organs and tissues that will ultimately form the baby, okay? So as you can see here, we've gone from a totipotent morula, where this is just one entity here, can give rise to everything, the extra embryonic tissues as well as the tissues and organs that will give rise to the baby. With further development, we're in the blastocyst stage here. These outer cells give rise to tissues such as the placenta, and these inner cells give rise to the organs and tissues of the baby. But now we refer to these inner mass cells, and we would the cells outside on the outside layer of the embryo as well, as being pluripotent. Okay, they're not totipotent anymore because these inner mass cells cannot give rise to tissues such as the placenta. Okay, and we're going to go over those terms in a bit more detail, but we'll just continue further along through this diagram. And so here we have the pluripotent inner mass cells of an embryo. These will eventually differentiate a little bit into multipotent stem cells, and the multipotent stem cells are the ones, for example, the heart stem cells will give rise to the cells that will form the muscle cells of the heart, as well as the cells that line the blood vessels, the cells that give the blood vessels their structure, for example. Okay, there will be a multipotent line of cells, nervous system stem cells that are multipotent, that will give rise to all the cells that are in the brain, and there are various types, as well as those that are in the nervous system. And again, there'll be another set of multipotent stem cells that will give rise to the cells that function within the immune system. Okay, so the end result, imagine you have a neural cell, a neuron, that's one single cell. It's an example of a nervous system cell. That would be unipotent, okay? A unipotent cell, that's a cell that cannot change into any other kind of cell, and it can't change back to a stem cell, at least not within the body. So it's sort of like the last stop. This is the final destiny of the cell, is, for example, in the nervous system, to become a neuron, and those types of cells are unipotent. And those are the ones you're probably most familiar with, is, you know, the outer cells of the epidermis of the skin. that would be an example of a unipotent type of cell. I mean, that's what its job is to be on the outer surface of the skin, and it's not going to revert back into any kind of stem cell. It's unipotent. So let's just review some of the terms we just went over and put them into a slightly different format that'll help you distinguish between them. So when we were talking about embryonic and adult stem cells, we've been talking a lot using this term potency. And potency is essentially the differentiation potential. So what is the power of that cell to differentiate into other types of cells? And we talked about totipotent stem cells, and this is a cell that can differentiate into any cell. So any cell at all. If it can differentiate into any cell at all, it's totipotent. A stem cell that is slightly more differentiated can differentiate, and this type of stem cell can differentiate into nearly all types of cells. These are called pluripotent stem cells. Multipotent stem cells can differentiate into a number of cells, but only those of a closely related family of cells. So adult stem cells tend to be multipotent. And the one thing missing from the table is the unipotent cell, and of course that's not a stem cell at all, and those are the cells that have fully differentiated into their final cell type. What I recommend you do right now is just pause the video for a few seconds and see how well you understand what we just went through. I have a table here where we have the type of cell, and I've given you some examples, and then you need to choose what is the potency level of that type of cell. Is it totipotent, pluripotent, multipotent, or unipotent? So as I said, just pause the video for just a couple seconds here and just jot these down before you move on. So up to this point, we've been talking about how multipotent adult stem cells that are naturally found in our bodies are available, but the only pluripotent stem cells are the embryonic ones that can form the baby. It used to be that the only way of obtaining pluripotent stem cells would be from embryos. And the other thing we've been saying all along is that once a cell is specialized, it can't go backwards and become a stem cell again. So essentially the only way of obtaining pluripotent stem cells would be from the inner cell mass of an embryo, and these are called HES cells. So these would be human embryonic stem cells. That's what it stands for. But in 2006 in mice, and then in 2007 in humans, a researcher called Shinya Yamanaka found a way of turning specialized adult cells from an adult body back into pluripotent stem cells, which were then able to give rise to different types of cells in the body. Now this was a completely new technique, and the cells that were produced are called iPS cells, and that stands for induced pluripotent stem cells. So here we are taking unipotent specialized cells in the adult body in the laboratory, turning them back into pluripotent stem cells, which have been demonstrated to then be able to develop into various specialized cells needed in the human body. What's happened is that these specialized adult cells, through a series of steps, have been genetically reprogrammed to an embryonic cell state. And they've been stimulated to express genes that are important for what I'm going to refer to as stemness. And essentially these are characteristics that give embryonic stem cells their stem cell abilities. Well, this is exactly what has been done to the human cells, is they've been stimulated to express genes that will make them like embryonic stem cells. So you see the importance again of gene regulation. Okay, so it's not that the DNA in the cells are different, it's how those genes have been regulated that will give the stemness characteristics of an iPS cell. Now the one thing that we do need to say is that they are different from human embryonic stem cells, and that's these HES cells that are shown here. And it is not yet known whether these differences are clinically significant. So there are are some differences, but this is just an enormous breakthrough because in the past, the only way of getting pluripotent stem cells was from the inner cell mass of an embryo, but now it is possible to get pluripotent stem cells from specialized adult cells. And as you can imagine, this caused a huge flurry in the scientific world. So here is a timeline running from 2006 to 2015. And you can see here, Yamanaka discovered that only 4 genes were needed to reprogram adult mouse cells into the embryonic-like pluripotent iPS cells. And these genes are critical to the cell regaining the stem cell characteristics, or the stemness characteristics. And in 2007, Yamanaka and James Thompson were able to do this in human cells. By 2009, there were more than 300 papers published on iPS cells, and this is just phenomenal. Lots of people started working on ways to create these iPS cells without inserting genes into the genome, because that was one of the things that was done early on, is that genes were inserted into the genome and the expression of genes was altered. Well, they started doing that without having to insert genes into the genome, and they were able just to change the regulation of the existing genes. In 2012, Yamanaka received the Nobel Prize, and in 2014, researchers in Japan began the first, and so far only, test of iPS-derived cells in people, and they were trying to treat a degenerative eye condition. Now, it normally takes 20 years to go from a discovery in the laboratory, at least 20 years, to clinical type trials. And you can see that the trajectory was much shorter here, far less than 20 years. And so it's probably no surprise that they had to halt the trial, because what happened is, as they began to prepare the next set of patients within this trial, they found 2 small genetic changes in the original patient's iPS cells. And they were worried about those. There was no evidence that these changes would have led to a tumor, but they decided to halt the trial to be on the safe side. So despite the fact that this was a disappointing outcome to the original discovery, what has been shown is that iPS cell-based therapies are definitely something that will be happening in the future, and certainly research in many areas is progressing along a lot of encouraging venues, okay? So just to reiterate, it normally takes at least 20 years to move from a scientific discovery to clinical trials and treatments being available. So you can see that this timeline was very, very short. And so while there has been a setback, still certainly there is a lot of promise in iPS research. Now in this slide, I just wanted to give you a sense of where we're at with respect to embryonic, adult, and iPS cells. And I've provided a few points for you to think about, not only within the context of this course, but after the course is over and you see news articles and you might be thinking, well, is this a good idea? Is this a not so great idea? How do I feel regarding these sorts of issues? And this is by no means an exhaustive analysis, and there are a lot of issues that we can't explore in great detail in this course, but I just wanted to touch upon some of them and kind of give you a sense of how to compare these different types of stem cells. Certainly in terms of the context of this course, on a final exam, maybe I might ask you, if you were to compare embryonic stem cells with induced pluripotent stem cells, what would be 2 advantages and 2 disadvantages of using one or the other type? So that's the kind of thing that I'm looking for in terms of the course, but I wanted to give you a little bit more in terms of your thoughts after you leave the course. So just to go over this very quickly, embryonic stem cells, they're pluripotent. We've discussed that. They can grow in large numbers in culture. Years of research have enabled us to direct their differentiation in very predictable ways. And in many ways, they are the gold standard for research into other types of stem cells. So whatever current or future stem cell technologies we have, they often will need to be compared with embryonic stem cells because they are the gold standard. Like an embryonic stem cell is the perfect cell, and so if we are going to try and use something other than embryonic stem cells, we need to make sure that they are safe and that they behave in ways we can predict. Certainly in the future, it may be possible to treat many diseases with stem cells originating from embryos. However, some of the disadvantages include immune rejection issues. If the stem cell came from another person, the embryonic stem cell came from another person, there can be immune issues. And of course, and I'm barely touching on this, there are complex ethical considerations that still remain. Adult stem cells are multipotent, so notice they're not pluripotent. One of the other disadvantages is they're rare in human tissues, and the cultural methods to generate large numbers of these is ongoing. But that's going to get better. There's an enormous potential for them for research and disease therapy. The likelihood of immune rejection issues are much lower compared with embryonic stem cells. For example, if you needed a new liver, in the future, it may be possible to take one of your liver stem cells and grow you a new liver, either within your body or outside of your body, but they're using your stem cells, so the likelihood of immune rejection is very, very low. Ethical considerations, because we're using adult stem cells, are simpler as well. And then induced pluripotent stem cells, they're pluripotent in many of the same ways as embryonic stem cells, but right now it is still not clear whether they behave exactly the same way as the embryonic pluripotent stem cells do. Okay, so that's why embryonic stem cells are still the gold standard for research and for comparison of new methods and technologies. A lot more research is needed, but it's extremely promising for drug discovery and disease model research, and we're going to talk about this a little bit more in the lecture. Again, extremely promising as a source of stem cells for disease treatment, because you can take an adult cell and induce it to become a pluripotent stem cell. We don't need to go hunting for the multipotent but rare adult stem cells, okay? But with the iPS cells, much more research is still required. Again, like the adult stem cells, likelihood of immune rejection issues are much lower, and ethical considerations, because these are coming from adult cells, are again much simpler. So what I recommend that you actually do with this video right now is just stop just for a few minutes and set yourself up a table just to summarize this information. This is sort of table like, something pretty simple just for yourself. You could put advantages down one column, disadvantages down another, and then the three types, the embryonic stem cells, the adult stem cells, and the iPS cells, and just fill out this table, because that'll help you for studying. Because like I said, you don't have to memorize everything on this table, but on the final exam I could ask you something along the lines of compare these two types of stem cells and give me 2 advantages or 2 disadvantages of that, and converting this material into a table will really help you learn the material in time for the exam. Now that we've discussed the key characteristics of embryonic and adult stem cells and also iPS cells, let's start talking about current stem cell therapies. Now, our first example is with respect to bone marrow transplants. We've touched on that a little bit already, and many of you have already heard about this. And where bone marrow transplants are often used is in the case of leukemia, which is a cancer of the blood or the bone marrow cells, and it's characterized by an abnormal increase of white blood cells. And one way of treating leukemia is to remove the cancerous blood cells using chemotherapeutics and or radiation. So essentially the cancerous cells are killed, but unfortunately at the same time any normal stem cells the person might have or any of the precursor cells that give rise to your blood cells, those are also destroyed. So this is a very bad situation, because essentially you can get rid of the cancer But at the same time, you kill the stem cells and the precursor cells in the bone marrow, and therefore the patient can no longer make new blood cells, so the patient would actually end up dying as a result of the treatment. So an overview of how bone marrow transplant therapy actually works is, in this example, we're showing a mouse. So here we have a mouse that had a leukemia. It had a radiation that killed the cancer cells, but it also halted the blood cell production. So if the mouse didn't get any other treatment, it would die. And so what happens here, and it's essentially the same idea in a human, is you then inject bone marrow cells from a healthy donor into the mouse. Over time, the injected stem cells colonize the hematopoietic tissues and allow the mouse to generate a steady supply of new blood cells, and this allows the mouse to survive. So how do we do this in a human who requires a cancer treatment that will destroy their blood cells? So there are essentially two blood stem cell transplant therapies that are well established. The first one is a bone marrow stem cell transplant. And what happens is the doctor, using a long needle, will remove bone marrow either from the cancer patient, him or herself, or from a donor, and it's this bone marrow that will be used in the patient after they've had their chemo or radiation therapy. The other method is called a peripheral blood stem cell transplant, and it's similar. In bone marrow, there are tons and tons of these blood stem cells, lots of them, very dense, in a dense population of them in the bone marrow. But in peripheral blood cell transplants, you have these hematopoietic stem cells in the circulating blood, and they're very similar to those in the bone marrow, but you don't have quite as many. Now, there are some techniques to get either the patient or the donor to grow more of these circulating hematopoietic stem cells, and essentially what happens is the blood from the patient or the donor is collected, and the hematopoietic stem cells are collected from within that larger blood sample. In both cases, unless the patient is using his or her own bone marrow or peripheral blood, an immunological rejection is possible. So that's why you always hear people talking about tissue matches. And if somebody needs to have either a peripheral blood stem cell transplant or a bone marrow stem cell transplant from someone else, if they can't use their own stem cells, then that patient is likely to be on anti-rejection drugs for the rest of his or her life. Now, it still saves their life, so it's still a good treatment, but still they have to take the anti-rejection drugs. Now, how this works in terms of determining whether a patient can use their own stem cells or whether they need a donor, in this case, if one of these cells is cancerous, well, in terms of the bone marrow transplant, you can take the bone marrow out, separate and purify the stem cells so you just get these ones. Then when there's radiation and chemotherapy treatments, all these cells will be destroyed, but you've already taken these ones out and purified them, and then you can put these purified stem cells back into the patient, and they will slowly grow and replace all the patient's irradiated cells, so these myeloid and lymphoid stem cells, as well as all the other blood cells that they need. So this is a case where a patient can use their own stem cells, so there's little chance of immunological rejection, and certainly having to take anti-rejection drugs is no longer an issue. On the other hand, if one of these cells is cancerous instead, then we cannot use the patient's own stem cells. This is a case where we need a bone marrow donor, and you need to match the donor with the patient. You need immunosuppressant drugs, and it's just much more complicated. So bone marrow transplants, either using the patient's own stem cells or those of a donor, whether it be through the bone marrow or the peripheral blood, is a very well established stem cell therapy and has been shown to be safe. However, when you need to have a donor, when you think about how hard it can be to find a donor match, you can certainly see the appeal of iPS technologies and why people were so excited when these were discovered. Another example of an established stem cell therapy again involves blood stem cells, and in this case, it's umbilical cord blood stem cell transplantation that we're talking about. And what happens is when a baby is born, the umbilical cord blood is obtained because the mother's donated her infant's umbilical cord and placenta after birth. And cord blood has a higher concentration of those hematopoietic stem cells than is normally found in adult blood. Now, there's a smaller quantity of blood in the umbilical cord, but there's a higher concentration of these hematopoietic stem cells. And what happens is those stem cells are frozen and stored. And when the baby grows up, if the baby needs those blood stem cells, then they can be made available. But they are also made available to others with the permission of the parents, and they're also used for ongoing research. And it has been found that there certainly have been fewer problems with rejection using and matching when using umbilical cord blood stem cell transplantation, and also the umbilical cord blood has been found to have stem cells that can be used for treatments for other diseases. So this is a second established and current stem cell therapy. So we've just done a couple of examples of current stem cell therapy. So where are we going in terms of research and future stem cell therapies? Well, first of all, stem cell research is extremely promising for basic cell biology. For example, with cultured pluripotent stem cells, whether they be embryonic, of embryonic origin, or iPS cells, well, they can certainly be used to study cell differentiation, and that is really important for understanding prevention and treatment of birth defects. So that's just some basic cell biology, and stem cells are extremely powerful for understanding certainly birth defects and how they might be treated. The other thing is that with cultured pluripotent stem cells, it's possible to use these stem cells to model diseases, so both genetic and other types of diseases. So we can use them to model diseases, and at the same time, we can identify drug targets and test potential therapeutics for those diseases, because you can imagine a situation where you have some pluripotent stem cells and maybe you want to study some kind of disease of the liver. Well, it would be very powerful to be able to create some liver cells, and even well before you begin testing the drugs in animals or humans, be able to quickly identify drug targets that work just in a Petri dish using cell lines. Extremely powerful method for testing drugs and modeling disease in general, because there's still so much we don't know about how various diseases develop, what happens at the molecular level in the cells, and so on. And certainly the drug discovery aspect of modeling disease is incredibly important, not just for genetic type diseases, to treat genetic type diseases or other types of diseases, but cancer is a big one here. You can also use stem cells for toxicity testing, so it is certainly a cheaper way and sometimes more effective way of testing the effect of various environmental toxins. The other thing that stem cells can be used for is certainly for future stem cell therapies. And the things we are talking about here are cell-based therapies in general. This is a very general term, and essentially this means any kind of therapy where you induce stem cells to differentiate into a cell type. And cell-based therapies certainly provide the foundation for tissue regeneration. And tissue regeneration is one aspect of the regenerative medicine that you may have heard about. Regenerative medicine is a fairly broad term that can involve treatment with stem cells, but also treatment with other things. But certainly tissue regeneration is a type of regenerative medicine. And the cell-based therapies that are being studied with cultured pluripotent stem cells certainly lay the foundation for potential tissue regeneration therapies in the future, either outside of the body or what seems even more amazing, but is certainly possible in the future, it may be that we can induce your existing adult stem cells to differentiate into a needed cell type without any kind of invasive procedure. And certainly the third future stem cell therapy is stem cell transplantation therapies. And that's an area that you've probably heard a lot about. It's been very prominent in many headlines. You've probably seen a lot of positive headlines and stories about stem cell transplantation therapies and negative ones. Certainly they're being offered in private clinics throughout the world. And the idea behind this is that you can grow tissues or cells for transplantation from these cultured pluripotent stem cells. For example, you could grow bone marrow cells for leukemia and chemotherapy. I mean, we already have an established technique for this, but you could grow more. You could grow nerve cells for Parkinson's and Alzheimer's disease, heart muscle cells for heart disease, pancreatic cells for diabetes, just to name a few. And this picture shows you probably why stem cells get so much play in the media. Is these are potential uses of stem cells, and these are just some of the potential uses of stem cells. And if you just look through this list of diseases, some of them are just so horrific, and the desire to create treatments for these awful diseases is really, really strong. And there's a huge potential of stem cells to really help with this, and it's really, really very exciting. But where are we at in terms of stem cell transplant therapies? I'll show you a slide about that. Here we have an example where we've got some mouse adult stem cells, and they've been injected into the muscle of a damaged mouse heart. Okay, so we're talking about a mouse here. So we're talking mouse adult stem cells injected into a mouse. Did it work very well? Did things get better for this mouse? Well, it turned out that stem cells did help to regenerate the damaged heart muscle. So these researchers went on and they took a mouse with some damaged heart cells, and then they injected human adult bone marrow stem cells into the rat and tried to see if they could induce new blood vessel formation in the damaged heart and make the heart function in the mouse a little bit better. The results of this study are very, very promising, and there have been a few small studies done in humans, usually those undergoing open heart surgery. So these tend to be very sick individuals. Very small scale. It's only been done on a few people, but there's a lot of promise, even though the mechanism is controversial. So why don't we just start using these cell transplant therapies on very, very sick individuals? Well, it turns out that they can be dangerous, and especially in these early stages, we really need to know a lot more about how the stem cells are likely to work in transplant therapies before they can become considered safe and widespread. And some of the things that people are working on, and there's a lot of research going on now, there are certain requirements to make a stem cell transplant therapy safe. The first one is we need to be able to grow enough of the stem cells to make a tissue and make enough for a transplant. And this needs to happen in a reliable way. And we're not there yet. We're getting there. A lot of people are studying that. But contrary to what you see in the headlines, there's still a lot of work to be done there. We need to make sure that differentiation of the stem cells into the desired cell types needs to be controllable and reliable. It needs to happen in a predictable way. We need to ensure that these transplanted stem cells survive in the recipient after transplant. A lot of the transplant treatments on offer, there is little evidence that those stem cells are surviving in the recipient afterwards. A lot more work needs to be done in this area. You need to make sure if you're going to transplant some stem cells into a patient, you need to make sure that those stem cells will integrate with the surrounding tissue after the transplant. We need the stem cells from the transplant to function for the recipient's lifetime. So these are important treatments. We need them to function for the recipient's lifetime because certainly, generally, stem cells do function for a recipient's lifetime. And last but not least, and this is where some of the controversy has been in the media, we need to make sure that the stem cell transplant or the therapy does not harm the recipient. Now that we're at the end of this video, you're in a much better position to evaluate stem cell research, certainly the stories that you see in the media. We've laid the foundation by learning what stem cells are, the differences between adult and embryonic stem cells, and how iPS cells fit into the picture. We've talked about established stem cell therapies and did an overview of current stem cell research that will undoubtedly lead to really, really exciting stem cell therapies within your lifetime. You now have the tools to better evaluate whether stem cell reports in the news are hype or hope, and in general, the potential of stem cells is so enormous and well beyond this course, you will need to critically evaluate their use, not only for yourself or for your family, but also as a citizen of the world. As always, if you have specific questions about stem cells, you should consult your doctor. But if you just want to find out more, both now and in the future after this course is over, we highly recommend visiting some Canadian websites because remember, Canada is a world leader in stem cell research. The first website is stemcellnetwork.ca. An alternative is the Canadian Stem Cell Foundation at stemcellfoundation.ca. There you'll find a wealth of reliable information regarding stem cell research and therapies.